Hi, I'm Scott Lindell, Research Specialist at the Woods Hole Oceanographic Institution and Lead Principal Investigator for a sugar kelp breeding project funded by the U.S. Department of Energy's ARPA-E Mariner program. Today, I'm going to give you a tour of our breeding project and the supporting facilities involved. Here at the University of Connecticut and in Woods Hole, we have collections of wild kelp from the northeastern United States. These consist of hundreds of gametophytes from nearly 18 locations and tested at 2 research farms. For the past 2 years, our team has been using these to make hundreds of unique single-pair crosses of gametophytes each fall for grow-out on research farms. Here's how the selective breeding process works. First, a quick reminder of the basic biology that is involved in kelp breeding. Every kelp blade starts from the fusion of eggs and sperm from female and male gametophytes, which are the microscopic alternative life stage that ultimately creates the blades or sporophytes. Our first task in a breeding program is to induce wild sporophytes to release spores that will become gametophytes. These are then separated mechanically into 96-well plates. Healthy, vigorous gametophytes are stepped up into ever larger vegetative or clonal cultures under red light. It can take up to a year to develop enough biomass for creating single-pair breeding crosses. About 6 to 8 weeks before we anticipate planting a farm, we induce our gametophytes to develop reproductive fertility by giving them an optimum environment. Then we introduce each unique male to a unique female gametophyte culture in order to create several hundred unique crosses. 2 weeks later, after they reproduce and develop sporophytes, we start the next stage of the nursery process. This requires applying the newly developed sporophytes from each cross to pucks, or pieces of PVC pipe that contain about 1 meter of wound seed string. We do this hundreds of times, so we eventually plant hundreds of 1-meter plots on the farms. The pucks are incubated in individual containers in a controlled environment, and after about 4 to 6 weeks, these pucks should look fuzzy, like a chia pet, with 1 to 2 millimeter sporophytes. That means they are ready to plant in the common garden test farms. Next, it's time to assemble the farm with the pucks and the grow lines. The trick to planting these lines lies in the preparatory work in the wet lab beforehand. There we carefully assemble the pucks into larger spools from which we can efficiently string consecutive plots onto the ropes. Then we unwind the string onto the grow ropes in the wet lab and place the ropes in coolers with seawater. At this stage, the puck strings are transformed to individual plots on the grow ropes. Then we move the coolers to the boat and prepare to reach the farm. Our common garden farm is an array of 5 parallel lines that are each 60 to 100 meters long and are closely spaced, just 1 meter apart. At the farm, the 5-line array is already set up. We simply attach each of the new grow lines, dump the cooler overboard, and then stretch the grow line out to attach the other end. Now it's time to sit back and wait. Our growing season is typically November to early June. Once late May or June arrives, we harvest the kelp before it starts to attract biofouling. We harvest each line, rope and all, and place it into insulated totes. Then these are trucked back to our lab in Woods Hole, where we can circulate 10 degrees C water through it overnight. The next day, we have staff working in pairs to evaluate every plot. First, we take measures of wet weight and blade density. Each 1-meter plot is hung up to weigh and then to snap a photo. Then subsamples are taken from the plot for enumeration of blade density and further analysis. Next, we complete the phenotyping of individual blades from the subsamples for each plot. The blades are laid side by side and measurements of each blade and stipe length and width and blade thickness are recorded. We record the condition of the blade, such as whether there are any fouling organisms or grazing, and whether the blades have source tissue. Each blade is individually weighed and subsequently dried in a greenhouse to record percent moisture. The dried tissues are analyzed for carbon and nitrogen, ash, sugars, and about 20 different ions and metals. So that concludes our tour. I hope you have a better understanding of the challenges involved in kelp breeding. Over the next year, we'll be publishing results of the important traits we've measured, many of which are highly heritable. We're very optimistic about the opportunities for improving kelp production through selective breeding, and we expect we'll meet our goal of achieving 10 to 20% improved yield per generation. I'm happy to take any questions now.